Okay, shall we start again? Well, we have to sometime, right? So, well, we'll see how long it takes to get through this lecture. Maybe 1 hour, 20 minutes, maybe a little bit less, to be honest. So the objective for this talk is to, well, explain to you how you can identify different bacteria, and then once you've done that, how can we classify them just like biologists do with any other type of organism. And in terms of classification, what you have in bacteriology is a kind of I don't know, kind of uneasy truce between two different approaches. So one of them, of course, is phonetic classification. That means that bacteriologists have been trying to classify bacteria for the last, 100 years or maybe more, way before the structure of DNA was discovered. So all of the, a lot of the classical bacteriological designations for species and the higher groupings, were initially done on the basis of, phenotypic characterization of bacteria. And then more recently, of course, now we can sequence whole genomes very, very quickly, get a lot of information about, genome sequences. So classification is done generally now by molecular phylogeny. Now, so these two kind of approaches coexist, and they've been synthesized by the, the big reference work in Bacterial Taxonomy, which is Bergey's Manual of Systematic Bacteriology. And the idea is that, all species will be grouped together in genera, and then several related genera in one family, then different families in an order, and so on. So the idea is to, get all bacteria to respect this, quite strict Linnaean hierarchy, okay, where you've got 1, 2, 3, 4, 5, 6, 7 levels in the classification. And then subspecies, you might have different strains of the same species of bacterium. And once we've done that, that's it. We've really organized those bacteria. We can be happy at, doing a good bit of work. So I'm going to talk first about the phonetic approach, how to identify bacteria based on their phenotypic characteristics, and then molecular phylogeny afterwards. So what characteristics can you use to distinguish different species of bacteria? Well, just like for macroscopic organisms, you've got like the visual phenotype. What do they look like? Either under the microscope or what their colonies look like. So under the microscope, you perform a gram stain, and you can determine whether you've got gram positives, gram negatives, cocci, bacilli, whether you've got streptococci, staphylococci, these types of information are a good start for identifying your bacteria. Whether they form endospores. If they form endospores, what's the location? What does the colony look like? Whether they have flagella or whether they don't. How are the flagella disposed or organized around the bacterium? So all of these are what you could call morphological characteristics. So I don't know, if you count how many things here, okay? Positive cell shape, so that's like 4 categories. Colonies, maybe double that up, endospores. So I don't know, if each of these are kind of binary thing, you've got like 2 to the 5 different categories here. So that's not enough, really. It's not enough character. There are not enough characters here just with morphology. So what are the other ways that you can characterize bacteria? So it's to do really with their physiology and their metabolism or their biochemistry. So this gives you more information. You've got more characters that you can use for identification and classification. So what does that mean? General nutritional type, heterotroph, autotroph, whether they use organic carbon or inorganic carbon. Carbon and nitrogen sources, what sugars can this bacterium utilize? What kind of nitrogen sources can be used, amino acids, ammonium or nitrate. Whether the bacteria are anaerobic or aerobic. And kind of related to the general nutritional type is how do they get their energy, whether they are thermophilic, mesophilic, whether they like acid, alkaline, or neutral pH. So these are, other elements of information you can use to characterize bacteria. And then for bacteria that are involved with other organisms, whether they're pathogens, whether they can infect, what kind of animals they can infect, what are their symbiotic relationships. So these are all part of a description of bacteria. So that's the kind of information we can get. And with just a few simple tests, you can you can begin to identify unknown bacteria. And this is what you're going to do in the lab classes, okay? Everything is going to start with an isolated colony on an agar plate. Because if you've got something that's mixed, a mixture of different bacteria, all of the other tests about their biochemistry are not going to work very well, because if you have, I don't know, a mix of bacteria and you want to find out whether one of the bacteria in the mix can use lactose. Okay, this culture is lactose positive. But if you've got 3 or 4 or 5 different bacteria in there, you don't know which one is lactose positive. So to do any of this stuff, you've got to start with an isolated colony in order to get information about the physiology of that organism. And if you don't have that, it's kind of a limit on what you can do. So start with your isolated colony, perform a gram stain, and you'll find out whether you've got gram positives or gram negatives, bacilli or cocci. And then according to what you see, you can perform some other tests. So if I've got bacilli, can they form spores? If so, well, they might be bacillus. If not, perform a catalase test. If they're negative, probably lactobacillus. If positive, maybe Listeria or Carinobacterium. And in fact, these two types have distinctive morphology, so you can tell the difference between them on the microscope. And then you have the same kind of thing over here. So if you've got gram-positive cookie, perform a catalase test. Streptococcus and Enterococcus are negative, Staphylococcus are positive. Just a simple test on the colony will give you a good orientation about what the identity of your bacterium might be. So same kind of thing with gram negatives. And then if your orientation test tells you, okay, I've probably got some kind of enterobacteria, what you're going to do in lab class is perform a whole set of biochemical tests in a kind of miniature format on an API gallery. So each one of these wells here has got a different kind of substrate. So here you've got different sugars, and this result is negative, this is positive. So, okay, this bacterium can use glucose mannose, but not inositol. So you've got a bunch of different biochemical characteristics that will help you identify what bacterium you have. So you have to remember that, okay, this is what you call an identification key. It gives you a number of tests to perform, very simple, and gives you an idea of what the bacterium might be. But it's not phylogenetic classification, all right? Yeah, if this was a phylogenetic classification, that would mean Staphylococcus and Streptococcus are very closely related and distantly related to Bacillus. Keep that in mind, and we'll find out whether that's actually correct later on. Okay, so they'll have characteristics in common, but this is not necessarily phylogenetic classification. So that's kind of classical approach to identifying bacteria in the lab. That's what you're going to do in the lab class. It's what probably was done in a clinical microbiology lab till about maybe 10 years ago. So it's kind of been a little bit, oh, well, not just that. I mean, there was a student who did licence pro, and he did his placement in a big company in the microbiology lab in a big food company, and they did this. This is what they did. Okay, on the tests for contamination of their different food products, they would use this classical type of bacteriology to detect the presence of bacteria. So it's still used, in microbiology labs every day, this kind of approach. Now, it's starting to be replaced by MALDI-TOF, which is matrix assisted laser dissociation ionization time of flight mass spectrometry. So once again, you have to start with an isolated colony, so you have a pure population of bacteria that are all identical. And then you pick off a bit of the colony and put it on the MALDI plate, which is a kind of metallic plate like this. And then the laser is going to come along and zap the spot of bacteria that you put on the plate. And it will vaporize some of the material that's on the plate and ionize it. So you get a kind of vapor of these ionized forms of whatever the molecules are that are in the sample. And then they get sent off to the mass spec detector, and this would generate a spectrum. Now, how on earth do you find out what this means? Okay, what you want to find out is, what was the bacterium in my colony? Well, the companies that make these machines, they spent a lot of time zapping hundreds of different strains of bacteria. So you have the name of the strain, and you have the spectrum which is associated with that particular bacterial species. So when the result comes back from the lab, it's going to compare the observed spectrum to the huge database that the company has of all the different spectra for different species of bacteria. And then once it finds a match, that tells you what your bacterium was. Okay, so why is this useful? Well, it's very fast. You can identify one sample in about less than 1 hour. And if you've got a machine that can handle a lot of samples, then you can identify the bacteria in the same day for 50 to 200 samples. Whereas for the classical approach, these kind of orientation tests, that's very fast. Okay, you do them on very quickly. You look in the microscope. That's 10 minutes, 15 minutes. These orientation tests, a couple of minutes. But this step here, putting the bacteria into culture with a battery of biochemical tests, then you have to culture them overnight. So you only get the result the next day. And that's the real advantage of this MALDI-TOF technique. So for a clinical lab, it might be very important to get the identification of the bacteria very fast. So the clinical bacteriologist can tell the hospital department, okay, your patient has got Staphylococcus aureus, or your patient has got Listeria, or your patient has got Shigella, or your patient has got, I don't know, whatever, okay? So having the result the same day can be very important to prescribe the right kind of treatment. So that's kind of like modern bacterial identification by MALDI-TOF. So these approaches will give you an identification down to the species level and tell you whether you've got yeah, Salmonella typhi, or it can tell you whether you have Escherichia coli. But in many cases, within a single species, you can have pathogenic strains and non-pathogenic strains, or strains which are very dangerous and strains which are, well, not quite so dangerous. So there is it is useful to have identification down to a subspecies level. So this can be, biochemical sometimes, whether they secrete a certain sugar. But most of the time, what you want to do is have a serological identification of the strain. And these are going to identify the K, the H, and the O antigens on the surface of bacteria. So what is very often done is to perform an agglutination test. So imagine you've got a bunch of bacteria which are, I don't know, serotype H7, okay? H7, H7, H7. What you do is you make a suspension of these bacteria in a little tube in water, and you mix it together with some beads, latex beads, which are coated with antibodies, which can be specific for different types of antigen on the surface of these bacteria. So if we've got an H1 antibody here, it's not going to react with the bacteria. So all of these bacteria will remain in suspension. However, if you take the same bacteria and mix them up with beads that are coupled to an antibody that's anti-H7, then these antibodies will bind to the surface of the bacteria, and it will all agglutinate together in a big mass. And so if you look at this, you look at the droplet of the suspension of bacteria and the latex beads. If you've got a negative result, where you just have a kind of uniform suspension of bacteria and the latex beads, whereas if you have a positive result, then you'll have clumps of bacteria and beads agglutinated together. So this is very fast. Okay, you get the reagents, you mix them together. You can have a panel of different antibodies that you put in a big strip, and you look at each one under the microscope. I don't know, just like 4x what's the word I'm looking for? Magnification. And then you can see visually whether you've got a positive result or a negative result. So this is a basis of serological tests by agglutination, very, very frequently used. So why do you want to do something like this, where you might want to determine, okay, imagine if we've got, I don't know, tomorrow morning, 25 students who've at the science faculty all come down with food poisoning, okay? And they are all infected with some kind of Escherichia coli. So, and you ask them, you say, oh, well, I had lunch at the canteen, I had lunch at the rue, and I ate, whatever and they all ate the same thing. Okay, so what's going to happen? You're going to send somebody to take a sample of that kind of food in the canteen, and you want to know if it's the same strain that infected everybody. So that's what you'll do. You perform your serological test, and if everybody's got the same serotype, and you find the same serotype in the food you suspect to be the source of the infection, well, that's pretty good evidence that all these people got infected by the same bacteria from the same source. So that's the kind of thing that this test is useful for, okay? Identifying the same bacterium in different situations. So identifying an epidemiological cluster. So the kind of molecular phylogenetic way of identifying strains of bacteria below the species level. There are different techniques now. So MLST, this is multiple locus sequence typing. So you're going to choose 5 or 10 genes that are present in all bacterial species, amplify them up by PCR, sequence them, and you get the sequence data. And if you have the same strain, they will have identical sequences. And depending on what taxonomic level you want to look at, you'll choose genes that are highly conserved or highly variable. Okay. So the other approaches are genetic fingerprinting. Not so sure if they're really used a lot anymore, but kind of interesting to know how they work. So there are repeated DNA sequences in many bacterial chromosomes. So ERIC is in Enterobacteria. Box, I think, is in repeated sequences in gram negatives and some gram positives. So what this is you have the bacterial chromosome like this. And somewhere in here you have these, so rep is repeated palindromic. So you have these repeated sequences distributed all the way around the chromosome. And the distribution of these repeated sequences is different between species, and it's different between strains of the same species. So what you want to do is use PCR primers that are specific for, one part of the repeated sequence. And so when you've got two that are close to each other, then amplify up the PCR product. Okay, for example, imagine these ones, they're kind of, these ones are too far apart. So in this example, I might have 1, 2, 3, 4, 5 different PCR products, which tells me something about the distribution of the repeated sequences. And then I'll take these PCR products and migrate them on a gel. So I'll have like this one, it's going to be a very small band. This one kind of quite similar size. 2 kind of medium-sized ones and 1 slightly bigger one. So for this bacterium, this will be my DNA footprint by rep PCR. So if I've got another strain of the same bacterium, doesn't have this repeated sequence here, this PCR product won't exist. So I'll get this kind of DNA footprint on a gel. So I'll be able to tell pretty quickly that I've got 2 different strains here. So that's how those ones work. Okay, so these are techniques for identification, distinguishing different bacteria one from another. Now, if you're a taxonomist, then you want to classify all these bacteria and get a phylogenetic tree, because this will give you information about the evolutionary history of different types of bacteria and how they're related to each other. So the first attempt to do this was from, phenotypic characteristics, whether they grow on this sugar, that sugar, gram positive, gram negative. And you want to try and translate this information into something numerical. So it's kind of a bit old-fashioned, but what would be done was that you would score each character as present or absent, and then compare pairs of bacteria. And for each pair, you calculate the kind of matching coefficient, okay? So this is the number of shared characteristics. This is the number of characteristics present in Y but absent in X. This is the number present in X, absent in Y, and this is the number absent in both of them. So from this matrix, you calculate some kind of number which tells you how close these 2 strains of bacteria are phenotypically. So if you've got, they're all identical in every respect. So this is going to be like 100, this might be 100, this is 0, this will be 0. Your matching coefficient will be 1. If they're entirely different, your matching coefficient will be 0. So the more similar they are, the higher this number is going to be, between 1 and 0. Now, probably not a good idea to group organisms together because they lack something. So I would say that probably this one is more a more valid comparison. Anyway, once you've done this, you can get a kind of similarity matrix. So once again, you've got, you know, your bacteria 1, 2, 3, 4, 5, 6, and this is the matching coefficient. So the ones that are, you know, the higher the matching coefficient, the closer they are. And then you can use distance methods to construct a cladogram here, cladogram. And this gives you your, you know, your taxonomic classification. So I tried to find some examples of this because it seemed to me pretty old-fashioned, okay? And I did manage to find a paper from 1994 where people did this to try and classify different species and different strains of Neisseria. So Neisseria gram-negative cocci in Diplococcus formation, and they're very important pathogens, some of them, for gonorrhea, okay, Neisseria gonorrhea, and Neisseria meningitidis, meningocoque. So they had all these different strains of bacteria, 31 different strains here that they wanted to classify. And of course, if you've got a lot of taxons, you need to have a large number of characters to compare to differentiate them. So here are all the tests that they performed on these bacterial strains. So they end up like with this massive kind of matrix of phenotype and bacterial identification and different bacterial strains. And in the end of it, so they got this as the phylogenetic classification of different strains of Neisseria, published in 1994, 25 years ago. Okay? And I must say that nobody does this anymore, but it kind of worked. There was a summary, the different techniques that can be used for phylogenetic classification of bacteria by molecular techniques. First one that was used was molecular hybridization. Okay, so you get the genome of one bacterium, you stick it on a filter, you get the genome of another bacterium, you label it radioactively, you fragment it, hybridize it on the filter, and you quantify how much of your radioactivity is retained. So if you've got two identical bacteria, the genomes are identical, right? So all the DNA here is going to hybridize on the filter. So you've got 100% is retained. The further apart they are genetically, the less radioactivity is retained. So this was the first, molecular method. And you can get good identification down to the genus and the species level. Genetic fingerprinting, depending on the kind of repeated sequences, genus, species, subspecies, multiple locus sequence typing. Actually, you can probably go up a bit here, okay, up to family, genus, species. Depends on which genes you choose. And two methods really which are perhaps the most useful, 16S RNA for, high-level taxonomic classification. And more you've got like whole genome approaches because it's become very much cheaper in the last 5 or 10 years. So more and more, no point fiddling around with sequencing 10 or 15 or 20 genes that you've chosen. Just sequence the whole thing and put it in the server to figure out the classification. So how do these two approaches compare? So I found like a similar paper on Neisseria published in 2012, kind of 20 years later. So they are trying to do the same thing as the previous paper, classify the different species of Neisseria. And this is multiple locus sequence typing, so they chose 52 ribosomal subunit genes, highly conserved proteins present in all bacteria. And they proved, in some other data or subsequently that in fact the classification they derived from these 53 genes was equivalent to what you get if you sequence the whole genome. So you don't get any extra information. So how does this shape up? Are we getting the same phylogenetic tree here? Okay, Neisseria gonorrhea, okay, they're together, that's okay. Same here. The meningitis group, okay, all together. Then you've got lactamica over here, cinerea over here. That's not the same, is it? Because here you've got cinerea phenotypically is kind of in amongst the meningitis group here. Whereas phylogenetic or bimolecular phylogeny, they're apart. So probably this is not correct in the sense that these non-pathogenic Neisseria cinerea species are probably not close at all to the pathogenic Neisseria meningitidis. So I think, you know, the molecular phylogenetics here is giving us an answer that makes more sense than trying to do all this phenotypic stuff. So this is an example of classification within one genus, right? You're classifying species within the genus Neisseria. So it works, okay? We're getting clustering of, you know, the pathogens, they cluster together, and all the non-pathogenic species, they cluster together. It's good. What kind of functioning correctly? So can you do this for all bacteria? Is it going to work? And we could just use these same 53 genes, sequence them, and it would allow us to get the phylogenetic tree for the whole of the bacterial world. And that would be really good. So I'm afraid the lines on this figure aren't really very visible. But the root is in here, and it's kind of, you can see all the lines here. Yeah, that's a bit unfortunate, actually, because some of the branching relationships here are important. I'll have to try and redo this figure and increase the contrast. Okay, so this is, let me just check, actually. It's within the sequence class Bacilli, yes. Okay, so this is molecular phylogenetic classification of the families within the class Bacilli. And it's working pretty well, okay, because all the strains of Streptococcus, they're clustered together. All the strains of Lactobacillus, they're clustered together. All the strains of Bacillus, they're together. Well, maybe not entirely, okay. Listeria, they're all going together. So it's working pretty well. And one of the things that you notice, in fact, is Staphylococcus is actually not very closely related to Streptococcus. So Streptococcus is probably phylogenetically closer to Lactobacillus, in fact, than it is to Staphylococcus. So what we saw from the identification key earlier on, where you're going to classify bacteria by their morphology and then catalase positive, catalase negative, looks like Staphylococcus and Streptococcus are quite close, but that's probably not the case phylogenetically. Okay, just thinking back now in evolutionary terms, if we think that Streptococcus is close to Lactobacillus, what is the ancestral state of the form of these cells? Coccus or Bacillus? Okay, try and remember this stuff from last year. What do you have at each one of these bifurcations? You have an evolutionary innovation. It occurs in one group, doesn't occur in the other group. So what I'm proposing here is you've got Streptococcus here. lactobacillus. And maybe over here you've got like a bacillus. And the character we're looking at is cookie bacilli, shape of the cell. What's the ancestral state likely to be? Yes. So what happened here? Yes. They lose something that gives them the elongated shape. And it's easy to lose because it's down to one gene. Exactly. So this might be deletion of MREB. Slightly wrong, MBL in gram-positives. So if you lose the gene that allows you to elongate, you return to becoming a cookie, right? So, yes, that's the answer I was looking for. Anyway, so different family within a class, multiple locus sequence typing, molecular phylogenetics, seems to give, consistent classification. Makes sense. So how about if we use it to classify all bacteria, okay? All eubacteria. So this is what you get if you use do the same thing on, well, hundreds of different strains of all types of bacteria. Does this make sense? Okay, so it's an unrooted tree, which you would expect because there's no outgroup really. Does it make sense? I'll let you have a little look, and what you need to look at here are the names of the groups and whether their position makes sense. Okay, oh yeah, okay, so the answer is here. Several inconsistencies. So can you see any inconsistencies? Well, yeah, it's true, you're not professional bacteriologist yet, so it's a bit difficult. Okay, look at the color green. Okay, color green, these are Clostridia. You've got a whole bunch of them over here. Fair enough. But you've got a whole bunch of them down here in a totally different clade. So this is not right for some reason. It's a problem. What else have we got? The other thing you can think about are the proteobacteria. So proteobacteria are, gram-negative rods, gram-negative bacilli. Okay, you've got like the gamma and the beta proteobacteria kind of close together. The alpha proteobacteria are way, way, way out on another branch. And then you've got the delta proteobacteria that are mixed up with a whole load of gram-positives around here. And the same thing with the epsilon proteobacteria over there. So that's not right either. That doesn't correspond to everything we know about the biology of these organisms. So it's not really working, okay? Trying to classify high-level classification with multiple locus sequence typing doesn't work all that well, really. So what's the other thing that is frequently used for phylogenetic classification of bacteria is 16S RNA sequencing. So this is a small subunit of ribosomal RNA. Now in bacteria, 16S, 23S, and 5S RNA are coded on the same operon. So they're the coding, the sequences coding for them are next to each other on DNA. And if you look at the 16S RNA gene, there are highly conserved sequences, which are in blue on this diagram, and variable sequences, which are in red here. So what you can do is choose PCR primers that hybridize to the highly conserved sequences, so they can amplify up the sequences from pretty much any bacterium. And then you can sequence the variable part that's in the middle. Okay, so it's a kind of perfect phylogenetic marker because all organisms have got, all bacteria have got ribosomes. The function is absolutely essential. And there's enough of a mix between conserved sequences and variable sequences for A, you can amplify them with the same primers, and B, you can get useful information from the variable sequences. So it's really very, very useful. So what you do is you amplify them up, sequence the variable regions, and then count the number of, the percentage of identity or the percentage of difference. So 25 years ago, in 1994, there was a publication saying, okay, I tested out 16S ribosome sequencing, and at that time they thought, okay, if we've got more than 97% identity, then we can say that these bacteria are members of the same species. More between 95% and 97%, they're members of the same genus. The same authors, or at least one of them anyway, revisited the same question 12 years later with a larger sample, and in fact the number goes up a little bit. So in fact we're talking about 98.7% identity means you're the same species. Well, what do you do with something that's 98.71% identical and 98.69% identical? There's always going to be a little bit of a kind of frontier here. But anyway, if you look at two very different types of bacteria, Salmonella and Bacillus, different different classes of bacteria, gram-negative, gram-plus, they only have 80% identity. So 16S rRNA sequencing is kind of universal mechanism of classifying bacteria. So does it work? Does it give us a useful classification? So this is from the textbook, and it gives you a summary of 16S rRNA phylogeny. And this is not so bad actually, okay? Because here we have all the gram-negatives, they're together, so all the proteobacteria are together, which wasn't the case before in the MLST that we saw. And up here you've got the gram-positives. And then the archaebacteria are separate. Now, so this looks reasonable, okay? So it's going to help us to answer some questions about the evolution of bacteria. So here you've got two sister groups, gram-positive and gram-negative. How do we find out which was the ancestral condition, gram-positive or gram-negative? Well, we need to find out whether these guys are gram-positive or gram-negative, right? And then we can infer which was the ancestral condition and which is the derived condition. Actually, I haven't memorized this, so I'm going to have to read my notes here. Okay, so Deinococcus thermus here seems to have two membranes, but no LPS. Chloroflexidae, gram-positive. do we have here? But Aquifax is gram-negative. Okay? So this one that's kind of at the root position is a gram-negative. So that means it's very, very difficult to tell from this tree which was the archaic condition. So they have an outer membrane, but it's not no LPS, it's not proper gram-negative. This is gram-positive. This is gram-negative. But if you look at the genome of this Aquifax genus, then you can see that it's a kind of a mosaic. It's had horizontal gene transfer from proteobacteria. And the same goes for Thermus, not Thermatoga. Oh, yeah, Thermatoga maybe, yeah, Thermatoga. It's also a kind of hybrid between eubacteria and archaea. And that means that the position of these organisms here is very difficult to tell whether they're really archaic or whether they go up here because of there's been too much lateral gene transfer. The other thing that's interesting about this is these are hyperthermophilic bacteria. They live in extreme environments like a lot of archaebacteria. So if that phylogenetic classification is correct, that might be telling us that the universal common ancestor up here, the origin of all cellular life, is coming or was living in a kind of thermophilic extreme environment, because you have the groups that are closest to the root that are still these thermophiles. So that's one way of looking at this. But there is meaningful information in the phylogenetic tree that can tell us something interesting about the origin of bacteria. And looking a bit further outside the 16S rRNA was really very important in proposing what's called the three-domain model of life. The idea that there's really three different types of cellular life: eubacteria, archaebacteria, and eukaryotes. And each of these three domains have very, very different types of 16S rRNA. And so often you can find like phylogenetic trees, universal phylogenetic trees based on 16S RNA sequences, and they show you, you've got eukaryotes down here. Oh, here we are. Archaebacteria and eubacteria. So if this is true, if this is a real, accurate representation of the history of life on the planet, then it's got some quite important consequences. One of them is, does it make any sense to talk about prokaryotes? Because prokaryotes are what? It's archaebacteria and eubacteria. But if these two groups of cells are so different, maybe we shouldn't put them in the same class. So there were some microbiologists who proposed this. We should stop using the term prokaryote. But most people, including me, think that we should carry on with this because it's useful, because they have a lot of similarities in simple cellular organization, the way they live, and it makes sense to set them apart from eukaryotic cells. The other implication here of this tree, if it's true, right, is look at the length of this branch here. So in a phylogram like this, the length of the branch is correlated with time. So if this is correct, that means that eukaryotes are really, really ancient, and they've been around from the beginning, just like bacteria. But that seems unlikely, to be honest. So there's kind of a problem with this, the idea that this tree is implying that eukaryotes are really quite different from all the other, from archaea, from eubacteria, and probably very, very ancient. Now, this idea changed maybe about 4 or 5 years ago with the publication in Nature of a discovery of another group of archaebacteria called Loci archaeota. which, well, let's say the sequences from the genomes of these bacteria were identified by taking samples down from an underwater volcanic vent in the far Arctic. And from the sequences, they got a bunch of sequences of conserved proteins from these loci archaeota, archaebacteria, which were very close to eukaryotic sequences. Okay, so here are the standard kind of archaebacteria, and these loci archaeota are very close to eukaryotes. What that's telling us here is that this tree might be false. And we get this false impression because the sampling of archaea is very, very poor. Up until, I don't know, 20 years ago, there were very, very few types of archaebacteria known. Once we increase the sampling, in fact, eukaryotes aren't really apart from archaea. But rather, it looks like the eukaryote sequences are very close to one type of archaea bacteria, like the loci archaeota. Now, another thing that's very interesting about the loci archaeota is that they seem to have a number of gene families that had previously been considered only present in eukaryotes. So these are eukaryote-specific genes. And these coded for things like, these escort proteins, they're involved in the formation of membrane vesicles. They're essential for forming complex membrane-bound structures inside the cell. So no other types of bacteria have these proteins, but the loci archaeota have the genes for them. Okay? They have the ubiquitin system, also specific to eukaryotes. So this was really a big thing, I think, anyway, because it's solving a little bit of the mystery of the origin of eukaryotes. So there's a previously unknown type of archaebacterium where a lot of genes are close to eukaryotic genes. And just this year, it's in preprint published on bioRxiv. There's a team in Japan who've actually been able to culture some loci archaeota. Because one of the criticisms about this paper, okay, is, yeah, you're basing all of this interpretation on a bunch of sequences that have been assembled by the computer. But how can you be sure you don't have some kind of eukaryote organism that's been mixed up with the sample, and you're mixing up the sequences and it's just a mistake? It's an artifact, okay? Not a real organism. So the only way to be sure about that is to get the bacterial cell, culture them, and sequence the genome. So this is what they've done here, these people in Japan. They sampled, sludge from a volcanic vent in the 2,500 meters underneath the sea in the Pacific. And over 12 years, okay, they characterized very slow-growing bacteria that came out of this sample. So they got this thing, which is here. It doesn't grow on its own, can't grow in pure culture. It needs to have one other bacterium together with it because it's got a kind of syntrophic, a symbiotic relationship. So they can grow the culture of two bacteria together and quantify the growth of each one. So the lag phase, you put it into culture for 1 or 2 months, it doesn't grow at all. Then when it starts growing, it takes like 3 weeks to double the numbers. I mean, this is, think about working on this project, right? You finish your PhD after 12 years. So these really slow growing, and it took, I think, I'm amazed that they carried on doing this, because it took a lot of, so you could almost like retire before you finished doing this. But anyway, the big thing here is that they managed to culture the first of these loci archaeota. So they can prove that, in fact, the genome really does belong to this cell. So it's a real archaebacterium. And they can have a look and see what's specific about them. So they have these cells that form kind of blebs like this. And you can see them in detail here. And sometimes they seem to form these long kind of membrane-bound projections. So they don't have any internal vesicles, but they do have these kind of long arms that are very unusual in bacteria. These are not flagella, okay? They're not pili. They're a kind of membrane-bound protrusion from the cell. So it might be what these escort proteins are doing, okay? They're involved perhaps, okay, maybe, in forming these weird structures. So this is going to be very, very interesting to find out more about the biology of these bacteria. And now that we have this organism here, we can start thinking about how this thing somehow met with proteobacterium and produced the first eukaryotic cells. Because this is the likeliest candidate, or something like this, is the likeliest candidate for the other partner in the endosymbiosis that led to the origin of eukaryotes. Okay, so that's kind of the end of the story here. Finishing very early. I'm so sorry about that. So there are some problems in bacterial taxonomy, okay, to determine bacterial species. The problem is there's no clear dividing line, okay? So, you know, for this kind of taxonomy based on sharing characters, the idea is, okay, if these matching coefficients are greater than 0.8, we can say they're the same species. For genome hybridization, the idea is if you've got more than 70% hybridization, they're the same species. For 16S rRNA sequencing, so you could you know, propose a dividing line, but there's always going to be some kind of limiting case where it's very, very close, and it's not going to be easy to determine whether you've got two bacteria that are the same species or slightly different. The other things that carry on being slightly problematic is that there are some inconsistencies with traditional names of bacteria and their phylogenetic classification. So Shigella and Escherichia, okay, so these are two genus level names for these bacteria. But in fact, in terms of their biology and their phylogeny, they're pretty much the same species, okay? Shigella is kind of like an enteroinvasive Escherichia coli. But, you know, it was just discovered 100 years ago by somebody else who gave it a different name. Clostridium difficile, or difficile. You know, pathogenic bacterium involved in a lot of hospital infections, difficult to treat. In fact, phylogenetically, it's not a Clostridium. So that's a big problem because every clinical lab in the world calls this thing C. difficile. So phylogenetically, it's supposed to be called Peptoclostridium difficile. And then some clinical biology said, no way, come on, this thing, C. difficile, everybody calls it that. So they had to change the name again. And it's now called Clostridioides difficile. Okay. So just to create a new genus for this bacterium and closely related species. So C. difficile is not Clostridium difficile anymore. Okay, so that's some slight problems in bacterial taxonomy. Really, the big problem is can we ever really understand what happened really something like 2.5, 2.8 billion years ago, the major divisions of life between archaebacteria and eubacteria. So the kind of traditional, the view that we want to have is that somehow we'll be able to derive a phylogenetic tree. This is the one true tree. It's like the Holy Grail. And once we find this true tree, we'll have we'll know how life evolved on the planet. Now, some people are rather pessimistic about this, including this evolutionary biologist William Ford Doolittle. So even 20 years ago, he was saying, look, the problem is the history of life. This tree is how we want to see things. We want it to be simple, okay, and we want it to be tractable by the human mind. But William Ford Doolittle said, well, maybe it's impossible, okay? Maybe, in fact, there's far too much lateral gene transfer in order for us to be able to get any clear idea. Okay, maybe up at the higher levels, we can understand the recent evolutionary history of groups of bacteria, but the further down we go, the more the signal is going to be hidden by these events of genes being swapped by different types of bacteria. So that's the problem with Aquifex and Thermatoga. They have part of their genome comes from one type of bacterium, part of their genome comes from another type. So where is the ancestor in this kind of mosaic? So that's a real problem. But, my feeling is that even if we can't know all the details, I think there is some useful information in this kind of classification, and that some of the big groups do have meaning. Okay, so I think I'm going to stop there. Actually, one of the lessons is I speak faster in English than in French, so when I give this lecture in French, it takes the right time, but today it's been a little bit too quick. So I'm going to stop there. If you have any questions, just ask me.